Hi, my name is Ahmad. I'm going to talk about the identification of bacteria, which possibly transmitted by Musica domestica in the region of Ahvaz, Iran. We're gonna we're gonna talk about the authors of these articles. There is two authors. First one called Babak, and the second one called Mahmoud. What I could only found few information for they both working in the Department of Medical Microparasitology School of Medicine and Infectious Diseases and Tropical Medicine Center. Ahvaz Chopar University of Medical Science. So they work on Ahvaz, Iran, on this school. Introduction. Houseflies have been implicated as important mechanical vector for lots of pathogenic microorganism agents, including bacteria, protozoa, worms, fungi, and viruses among humans and animals. Housefly can pick up microorganisms on different parts of its bodies, like hairs, body, appendage, and mouth parts. Houseflies' secretions regurgitate and feces can transmit the disease. Diseases such as food poisoning and dysentery regarding to bacteria. Therefore, housefly is recognized as potential vector of foodborne pathogens. The aim of the project, the aim of this articles was to isolate and identification bacteria that are picked up by housefly over the human and animal premises. This is the aim of this project. Material and method. A total 230 houseflies were collected from the central slaughter house and a zoo in Ahvaz, Iran. The samples were transported to the parasitology laboratory and placed in the sterile dishes in freeze temperature for 15 minutes to anesthetize them. The protocol procedure was followed to prepare the samples for culturing on blood agar media and differentiate media. The samples were washed by distilled water and then the flies were soaked in saline solution. Finally, the yield solution was transferred to a culture media by a sterile loop under hood and sterile conditions. The result of this study, from 230 houseflies, a total of 629 samples were positive, or isolated bacteria from housefly are pathogenic. This is the result. This is the diagram for the isolated bacteria from the housefly. They were isolated for kind of bacteria. The most present one was the E. coli. We see here the E. coli, and it was present in 36.5%. Then Staphylococcus aureus, it was present in 26.9%. And then Pseudomonas species, it was present in 21.8%. And the last present of bacteria was for Proteus. It was present in 14.8%. Like I said, these all were isolated from housefly. The discussion now. These results agree with the results of Silman in Malaysia. The specimen was collected from poultry farm. They found Idsntification species, Bacillus species, Enterobacter species, Proteus species, Escherichia species, and Klebsiella species. There are more studies which confirm the role of houseflies on transmission of different bacteria as a worldwide agent. The more evident to find the role of houseflies on the transmission of bacteria is using different insect species and different methods of transmissions. Olsen and Hamak isolated Salmonella enteritidis, Salmonella infantis, and Salmonella Heidelberg from houseflies over poultry houses. Nesny have isolated Bacillus species, Staphylococcus species, and Micrococcus species from feces and vomits of houseflies too, other than from the internal body of housefly. In the other study of Fotidar showed the vector potential of housefly in the transmission of Vibrio cholera in an outbreak in India. The conclusion now. The results of this project and other researcher study confirm that flies can pose potentially serious health risks as mechanical vectors. Therefore, they have to be controlled and density of their population should be reduced, undertaking different vector control approaches. The reference, this is the article reference, the one I used in my first presentation, and that's all what I have, and thank you so much for listening, and please ask if you have any question. Thank you so much. Bye bye.